You are stranger than you think. You are the only primate that walks upright on two legs. The only one with a layer of subcutaneous fat beneath your skin. The only one that can consciously control your breathing. The only one whose babies instinctively hold their breath underwater. The only one that is instinctively drawn to the sea. Every other great ape is covered in thick body hair and terrified of deep water. You are nearly hairless and you swim. The question is, why? Before we get down to business, please subscribe to this channel. Your support really helps us to continue this work. Thank you. Start with a chimpanzee. Not a cartoon chimpanzee, not a performing chimpanzee in a circus, but a wild, adult chimpanzee in the forests of Central Africa, doing what chimpanzees do. It is covered in thick, dark hair from crown to ankle. It moves on all fours when traveling any distance, knuckle walking with its weight distributed across both hands and both feet. When it encounters a stream, it wades through cautiously, keeping its head well above the water, visibly uncomfortable with anything deeper than its knees. It does not swim. It cannot consciously hold its breath. If you submerged a chimpanzee infant in water, it would not instinctively hold its breath and begin paddling. It would panic and drown. The chimpanzee is our closest living relative, sharing approximately 98.7% of our DNA. And in almost every visible, physical, behavioral characteristic related to water, it is our opposite. Now consider yourself. You are, by the standards of the primate order, extraordinarily strange. Your body is nearly hairless, retaining thick hair only on your head, in your armpits, and in your pubic region, a pattern of hair distribution that has no good parallel in any other primate, and that has resisted convincing evolutionary explanation for over a century. Beneath your skin, you carry a continuous layer of subcutaneous fat that no other primate possesses. A layer that gives the human body its characteristic, smooth, rounded surface, and that serves as insulation and energy storage in a way that is far more reminiscent of a seal or a dolphin than of a gorilla or an orangutan. Your larynx sits lower in your throat than in any other primate, a feature that enables complex speech, but that also, incidentally, allows you to breathe through your mouth and to take the deep, controlled breaths that skilled swimmers and divers use. You produce tears when you are emotionally distressed, a behavior observed in no other primate. And the closest functional analogues in the animal kingdom are the salt-excreting tears of marine reptiles and the lacrimal secretions of certain marine birds. And when you place a human infant, weeks or months old, in water with proper support, it will close its glottis, slow its heart rate, and begin rhythmic paddling movements, a response called the diving reflex that is not learned, not taught, and not present in the infants of any other primate. These are facts. They are not disputed. What is disputed, fiercely and for more than 60 years, is what they mean. The standard explanation, the one you will find in most textbooks and most popular accounts of human evolution, is that these features evolved on the African savanna in response to the demands of an upright, bipedal, tool-using lifestyle in an open grassland environment. The hairlessness was for thermoregulation in the hot sun. The subcutaneous fat was energy storage. The descended larynx was for speech. The tears were a byproduct of some other physiological change. The diving reflex is a vestigial remnant of something much more ancient, shared with all mammals, not specifically human. Each feature has an explanation. But a marine biologist named Alistair Hardy, sitting in his office at Oxford University in the 1950s, looked at this list of explanations and thought, none of them are very convincing. And he had a different idea. Alistair Hardy was not a fringe scientist or an amateur theorist. He was one of the most respected marine biologists in Britain, a fellow of the Royal Society, a professor at Oxford, a man whose scientific credentials were impeccable and whose judgment was trusted by his colleagues. In 1930, while working on a research voyage in the Antarctic, he had an idea that he described later as coming to him suddenly and completely. A vision of a population of early hominids retreating to the sea. He wrote it down. He thought about it carefully, and then he put it away for 30 years, because he was certain that if he published it, it would be dismissed as unscientific speculation and would damage the career he had worked his entire life to build. He kept the idea to himself for 3 decades, mentioning it only to a few trusted colleagues, until 1960, when he finally presented it publicly in a lecture to the British Sub-Aqua Club, and then published a brief paper in the New Scientist. The idea was this. Between 5 and 8 million years ago, the ancestors of modern humans diverged from the ancestors of modern chimpanzees. The standard account holds that this divergence happened when the forests of Central Africa fragmented, forcing some populations of forest-dwelling primates out onto the open savanna, where they gradually adapted to terrestrial life, developing bipedalism, losing their hair, and eventually evolving larger brains. Hardy proposed an alternative. He suggested that one population of ancestral hominids, rather than moving onto the savanna, moved to the coast. That they spent a significant period of their evolutionary history in and around shallow coastal waters, wading, swimming, diving for shellfish and other aquatic food sources, and that the time they spent in this semi-aquatic environment left permanent marks on the human body that are still visible today in our unusual anatomy. He called it the aquatic ape hypothesis. The reaction from the paleontological establishment was, as Hardy had feared, largely dismissive. The hypothesis was not taken seriously as a scientific proposal. It was treated as an amusing curiosity, an interesting if eccentric idea from a distinguished scientist who had perhaps strayed too far from his area of expertise. There were no fossil hominids from coastal environments. There was no direct evidence that early human ancestors had spent significant time in or near water. The idea felt, to most specialists, like motivated reasoning, taking a list of anatomical curiosities and constructing a just-so story to explain them in a way that was more poetic than scientific. Hardy's hypothesis might have faded quietly into the footnotes of the history of evolutionary biology if it had not been rescued, expanded, and argued with tremendous force and eloquence by a Welsh writer named Elaine Morgan. Morgan was not a scientist. She was a television writer, a highly successful one, but a person without academic credentials in biology or paleontology. She encountered Hardy's hypothesis in the early 1970s and found it, in her words,
the most persuasive evolutionary explanation for human uniqueness she had ever encountered.
In 1972, she published The Descent of Woman, a book that challenged both the aquatic ape hypothesis's neglect and the male-centered assumptions of standard evolutionary narratives, and followed it with a series of increasingly detailed and technically sophisticated books arguing for the aquatic ape hypothesis, culminating in the aquatic ape hypothesis in 1997. Morgan was a brilliant polemicist and a careful researcher, and she engaged the scientific literature with a seriousness and depth that belied her lack of formal credentials. She also had the gift, rare even among professional scientists, of being able to explain complex anatomical and physiological arguments in language that ordinary readers could follow and find compelling. Morgan's central argument was simple and powerful. The standard savanna theory of human evolution had been accepted not because it explained human anatomy well, but because it was the only theory on the table. When you examined each of the distinctive human anatomical features that distinguished us from other primates, and asked whether those features made more sense as adaptations to savanna life or as adaptations to a semi-aquatic existence, the aquatic explanation was consistently more parsimonious, more complete, and more convincing than the savanna explanation. Not for every feature, not overwhelmingly for any single feature, but across the full pattern of human anatomical distinctiveness, the aquatic interpretation kept turning up as the more elegant explanation. And that pattern of consistently better fit was, Morgan argued, itself significant. Let us go through the evidence carefully, because it deserves to be examined on its merits rather than dismissed on the basis of its unconventional origin. Start with subcutaneous fat. Human beings have a continuous layer of fat directly beneath the skin that is absent in other primates. Chimpanzees, gorillas, orangutans and gibbons store their fat in concentrated deposits around internal organs, as is typical for mammals generally. Humans store fat in a subcutaneous layer that covers most of the body surface, giving human skin its characteristic smooth, padded quality and providing insulation at the body's surface. This subcutaneous fat distribution is precisely the pattern found in aquatic and semi-aquatic mammals, from seals and dolphins to hippos, where it functions both as insulation in cold water and as a buoyancy aid. It is not the pattern found in other savanna-dwelling mammals, which do not need surface insulation. The savanna explanation for human subcutaneous fat, that it serves as energy storage, fails to explain why the fat is distributed subcutaneously rather than around internal organs, as in all other primates. The aquatic explanation, that subcutaneous fat evolved as insulation and buoyancy in a water-using ancestor, explains both the presence and the distribution of the fat in a single parsimonious account. Human hairlessness is perhaps the most visually striking way in which we differ from other primates, and it is the feature for which evolutionary explanations have been most vigorously contested. The standard account is that bipedal savanna hominids lost their body hair because an upright posture reduced the body surface exposed to the overhead sun, and because a hairless body loses heat faster through sweating than a hairy body, making thermoregulation more efficient during sustained running and foraging in hot, open environments. This explanation has some merit, but it has problems. Other savanna mammals, including lions, hyenas, baboons, and zebras, are not hairless, suggesting that savanna life does not reliably select for hairlessness. Large savanna mammals that are hairless, primarily hippos, rhinos and elephants, are all either semi-aquatic or descended from aquatic ancestors, a pattern that Morgan found highly suggestive. Human hair loss is also incomplete in a way that is consistent with water exposure, but harder to explain as a thermoregulatory adaptation. The areas where hair is retained, the head, the armpits, and the pubic region, are precisely the areas that would be above water or protected from current drag in a wading or swimming animal. The head hair in particular, which is retained and which grows continuously in humans in a way unique among primates, has been interpreted by aquatic ape proponents as insulation and UV protection for the head, the part of the body most exposed to the sun when wading in shallow water. But perhaps the most compelling anatomical evidence for a significant aquatic phase in human evolution is the one that is hardest to explain away: the diving reflex. The diving reflex is a suite of physiological responses that occur when the face of a mammal is immersed in cold water. Heart rate slows, sometimes dramatically. Blood is shunted away from the extremities toward the vital organs. The glottis closes, preventing water from entering the lungs. In trained human divers, these responses can be developed to remarkable degrees, enabling extended breath-hold dives to depths of several hundred meters. But the key point for our purposes is that the diving reflex is present in human infants from birth, before any learning or training has occurred, and it is significantly stronger in human infants than in adult humans, suggesting that it is an innate response rather than a learned one. Human infants placed in water with their face submerged will automatically slow their heart rate, close their airway, and make swimming movements. This response is not present in chimpanzee infants. It is not present in gorilla infants. It is not present in the infants of any other non-human primate. It is present in the infants of diving mammals, including seals and otters. And it is, in human infants, a response that fades with age unless it is reinforced by regular water exposure, which is precisely the pattern you would expect from an adaptation that was important in ancestral environments, but is no longer regularly activated in modern human development. The conscious control of breathing that humans possess, and that other primates do not, is closely related to the diving reflex, and equally difficult to explain in purely savanna terms. All mammals have some capacity to briefly override their automatic breathing rhythms, but humans have an unusually well-developed ability to voluntarily control the depth, rate, and timing of their breaths. This ability is the physiological foundation of both human speech and human diving. Producing the complex rhythmic sequences of sounds that constitute spoken language requires precise voluntary control of the respiratory muscles that no other primate possesses to anything like the same degree. And that same voluntary respiratory control is what enables human divers to take deep, controlled breaths before a dive, and to extend the duration of a breath hold far beyond what involuntary breathing alone would allow. The aquatic ape hypothesis proposes that voluntary breathing control evolved primarily in the context of aquatic breath hold diving, and that complex speech was a secondary benefit of a pre-existing respiratory capability rather than the primary selective force that drove its evolution. This brings us to the descended larynx. In humans, the larynx sits significantly lower in the throat than in any other primate. In newborn humans, the larynx is positioned high, close to the nasal passage,
as it is in adult chimpanzees and other primates, which is why newborn human infants can simultaneously breathe and swallow, something adult humans cannot do.
The larynx descends to its adult human position during the first few months of life, in a developmental process that appears to be unique to our species. The descended larynx is generally explained as an adaptation for speech, creating the large pharyngeal resonating chamber that gives human vowel sounds their distinctive acoustic properties. But the descended larynx also creates the anatomical arrangement that allows humans to breathe through their mouths as well as their noses, to take the large controlled breaths used in diving, and to use the Valsalva maneuver to equalize pressure in the ears and sinuses during descent. Aquatic ape proponents argue that the descended larynx makes more sense as a primary adaptation for controlled diving breathing, with complex speech as a secondary consequence, rather than as a primary speech adaptation that incidentally enables diving. The question of human tears is smaller in scale but potentially significant in principle. Humans are the only primates that produce tears in response to emotional distress. Other primates produce tears as a lubricant for the eyes and as a response to physical eye irritation. But the copious emotional tears that flow down human faces during crying are uniquely human. The function of emotional tears has been debated for years without clear resolution in a purely terrestrial evolutionary context. Morgan argued that the closest functional analogues to human emotional tears are the lacrimal secretions of marine animals such as sea turtles, marine iguanas, and certain seabirds, which excrete excess salt through their lacrimal glands. She proposed that human emotional tears might be a vestigial salt excretion mechanism from a period when our ancestors were consuming large quantities of salt from marine foods and needed an additional excretion route beyond the kidneys. This is a speculative argument, and most researchers who have considered it regard the evidence as too thin to be compelling on its own. But as one element in a pattern of features that collectively point toward a marine or semi-marine past, it is, at minimum, interesting. Bipedalism, the feature of human anatomy that is in many ways the most distinctive and the most difficult to explain, is also addressed by the aquatic ape hypothesis, in a way that some researchers have found more parsimonious than the standard savanna account. The savanna explanation for bipedalism is that walking upright on two legs was more efficient than knuckle walking for covering long distances on open ground, freed the hands for tool use and carrying, and reduced the body surface exposed to overhead sun. These are plausible selective advantages, but they are not overwhelming, and the transition from quadrupedal locomotion to bipedalism involves a prolonged intermediate phase during which neither form of locomotion is fully efficient, which creates the standard evolutionary problem of how natural selection can favor a trait that is initially disadvantageous. The aquatic explanation for bipedalism notes that wading in shallow water naturally produces an upright posture, since the buoyancy of the water reduces the weight-bearing demands on the skeleton and makes an upright stance more comfortable than it would be on land. An animal that spent significant time wading in chest-deep or waist-deep water would find an upright posture naturally selected for, and the transition to bipedalism would be far less problematic than it would be on land, because the water provides the mechanical support needed during the transitional phase when the musculoskeletal system is not yet fully optimized for upright walking. This argument does not require that our ancestors were fully aquatic, only that they were regular waders in shallow water, precisely the semi-aquatic niche that the aquatic ape hypothesis proposes. The geological context in which the aquatic ape hypothesis has been placed by its modern proponents is worth examining in detail, because it addresses one of the most persistent criticisms of the theory: the absence of fossil evidence for hominids in coastal or aquatic environments. The region of Africa where the earliest hominid fossils have been found, the Ethiopian Afar region, was during the relevant period, roughly 5 to 7 million years ago, a dramatically different landscape from what it is today. Geological and paleoclimatic evidence indicates that the Afar region was periodically flooded by the sea, creating a large inland sea or a series of coastal lagoons and marine inlets that would have separated the Danakil highlands, a highland plateau in what is now Eritrea and Ethiopia, from the African mainland. This Danakil isolation scenario, developed in detail by the Belgian geologist Koen Martens and others, proposes that a population of ancestral hominids was isolated on the Danakil highlands when the surrounding lowlands flooded, and that this isolated population, cut off from the forest resources of the mainland, adapted to the coastal and shallow water resources that were available to them on the island-like Danakil highlands. The isolation would explain why aquatic adaptations evolved specifically in the hominid lineage, rather than in other African primates, and the subsequent re-exposure of the Danakil highlands as sea levels dropped would explain the return to terrestrial life and the transition to the fully bipedal, upright walking hominids that appear in the fossil record. This geological scenario is not universally accepted, and the evidence for a complete marine flooding of the Afar region during the relevant period is subject to ongoing interpretation. But it is geologically plausible, and it addresses what would otherwise be a significant weakness in the aquatic ape hypothesis. If semi-aquatic life was so advantageous for our ancestors, why did it not produce similar aquatic adaptations in other African primates? The isolation scenario provides an answer. It happened to one population in one place at one time, which is precisely how evolution typically works. The fossil evidence for early hominid use of aquatic and coastal food resources has grown substantially since Hardy first proposed the aquatic ape hypothesis. And while none of it is direct confirmation of the hypothesis in its strongest form, it provides a consistent backdrop of coastal and semi-aquatic ecology against which the hypothesis becomes more rather than less plausible. The site of Pinnacle Point in South Africa, excavated in the early 2000s, produced evidence of shellfish consumption by anatomically modern humans dating to approximately 164,000 years ago, during a period of severe glaciation when inland food resources were scarce. This is far earlier than the previously accepted evidence for systematic marine resource exploitation, and suggests that the relationship between the human lineage and the sea is far older than was recognized even recently. More significantly for the aquatic ape hypothesis, sites in East Africa have produced hominid fossils and associated food remains from lakeside and riverine environments, consistent with a long evolutionary history of close association with water-based food resources. The Homo ergaster and early Homo erectus populations that spread out of Africa beginning approximately 2 million years ago show in their anatomical features the full expression of the hairlessness, subcutaneous fat distribution, and bipedalism that the aquatic ape hypothesis seeks to explain. Their fossil remains are found in association with both savanna and riparian environments, consistent with a population that had recently emerged from a semi-aquatic phase and was in the process of adapting to a wider range of terrestrial habitats while retaining the anatomical features that had been shaped by their aquatic ancestry. The scientific community's response to the aquatic ape hypothesis over the decades has been more nuanced than a simple story of establishment rejection might suggest, though the nuance has not always been visible in the public debate. The mainstream of paleoanthropology has consistently rejected the hypothesis in its strong form, the claim that a specific semi-aquatic phase was the primary driver of the anatomical features that distinguish humans from other primates. The reasons for this rejection are both evidentiary and methodological. There is no direct fossil evidence of a semi-aquatic hominid population. There are no hominid fossils from coastal Miocene deposits in the Afar region. There are no anatomical features of the human skeleton that unambiguously indicate an aquatic ancestry in the way that, for example, the vestigial hind limb bones of whales unambiguously indicate their terrestrial ancestry. Each of the anatomical features that the aquatic ape hypothesis points to as evidence of aquatic ancestry has an alternative explanation that does not require a semi-aquatic phase. And most specialists consider those alternative explanations more parsimonious given the overall fossil record of human evolution. But the rejection has not been complete, and the alternative explanations have not been entirely convincing. A significant conference on the aquatic ape hypothesis was held in Valkenburg in the Netherlands in 1987, bringing together proponents and critics for a sustained scientific exchange. The proceedings of that conference, published in a volume called The Aquatic Ape, Fact or Fiction, revealed something important. The critics were more effective at challenging specific claims of the hypothesis than at defending the savanna theory against the same scrutiny. When aquatic ape proponents turned the questioning around and asked critics to explain specific human anatomical features in savanna terms, the answers were often as speculative and as incomplete as the aquatic explanations they were criticizing. The standard savanna account of human evolution had been accepted not because it comprehensively explained human anatomy, but because it was the dominant paradigm, and dominant paradigms are not held to the same evidentiary standard as challengers. This insight was developed most sharply by the paleoanthropologist Philip Tobias, one of the most respected figures in the field, who, at a 1999 conference in Ghent, made what was for the establishment a startling admission. The savanna hypothesis of human evolution, the story that our ancestors left the forests and walked out onto the grasslands and became bipedal in response to open ground, was not supported by the fossil and geological evidence. The early hominids were not savanna animals. The environments in which the earliest bipedal hominids are found, the environments in which Australopithecus and the earliest members of the genus Homo lived, were not open grasslands, but mosaic environments of woodland, wetland, and waterside habitats. The savanna hypothesis was not just inadequate, it was, in Tobias's assessment, factually wrong, which meant that the primary rival explanation to the aquatic ape hypothesis had been shown to be inadequate, and the aquatic ape hypothesis was now competing not with a well-supported mainstream account, but with a vacuum. The response of paleoanthropology to this situation has been not to embrace the aquatic ape hypothesis, but to develop what is sometimes called the waterside hypothesis, or the literal theory of human evolution, a more cautious and more specifically evidenced version of the basic aquatic ape insight. The literal theory, developed and argued by researchers including Michael Crawford, Stephen Cunnane, and Mark Verhagen, proposes that the distinctive features of human anatomy and cognition cannot be fully explained without reference to a long evolutionary history of association with water-based food resources. The theory is less committed than the aquatic ape hypothesis to a specific semi-aquatic lifestyle, and more focused on the nutritional and ecological consequences of eating marine and freshwater foods over an extended evolutionary period. The nutritional argument for a waterside theory of human evolution has become, in the past 2 decades, one of the most interesting and empirically grounded aspects of this entire debate. Human brain growth during the period of human evolution is extraordinary and biologically expensive. The human brain, which is approximately 3 times larger relative to body mass than the brains of other great apes, is composed primarily of fat, and specifically of long-chain polyunsaturated fatty acids, particularly docosahexaenoic acid, or DHA. DHA is found in high concentrations in marine foods, particularly in oily fish, shellfish, and seaweed, and is present only in low concentrations in the terrestrial plant and animal foods that would have been available to savanna-dwelling hominids.
The argument made by Crawford, Cunnane, and others is that the dramatic expansion of the human brain during the Pleistocene would not have been nutritionally possible without access to marine or freshwater DHA sources, and that the consistent presence of early human ancestors at lakeside, riverine, and coastal sites is not incidental, but reflects a nutritional dependency that was central to the evolution of human cognitive capacity. This is a more modest claim than the aquatic ape hypothesis in its original form, but it is better supported by evidence, and it leads to the same basic conclusion: that the relationship between the human lineage and water was not peripheral, but fundamental. The most remarkable living evidence for rapid human aquatic adaptation comes from a population that most people in the industrialized world have never heard of: the Bajau people of Southeast Asia. The Bajau are a maritime nomadic people who have lived at sea for centuries, diving for fish and shellfish in the waters of the Philippines, Malaysia, and Indonesia. Traditional Bajau divers spend a significant portion of each day underwater, diving without equipment to depths of 20 or 30 meters, spending 8 to 10 hours a day in the water in some communities. In 2018, a research team led by Melissa Ilado published a study in the journal Cell, showing that the Bajau have significantly larger spleens than their non-diving neighbors, the Saluan people of the same region. The spleen stores red blood cells and contracts during diving to release them into the bloodstream, increasing the blood's oxygen-carrying capacity in a response called the spleen contraction reflex. Larger spleens mean more oxygen stored, meaning longer and deeper dives. The Bajau do not simply train their spleens to be larger.
Their genetic profile shows variants in a gene called PDE10A that are associated with larger spleen size, variants that are found in the Bajau at significantly higher frequency than in the neighboring Saluan population. In the centuries since the Bajau adopted their diving lifestyle, natural selection has detectably changed their genome in a direction that makes them better divers. evolution toward aquatic adaptation happened in a human population, in response to water use, in a few thousand years. This finding is important for the aquatic ape debate in ways that go beyond its direct content. The standard criticism of the aquatic ape hypothesis has always been that the time required for the anatomical changes it proposes, the loss of body hair, the development of subcutaneous fat, the descended larynx, the diving reflex, would have been so great that a specific semi-aquatic phase would need to have lasted millions of years to produce visible results. The Bajau finding suggests that meaningful genetic change in response to aquatic adaptation can occur on a timescale of thousands of years rather than millions. Bajau spleen enlargement is a relatively minor trait compared to the full suite of changes the aquatic ape hypothesis proposes, but it demonstrates the principle that human populations are capable of rapid genomic adaptation to aquatic environments when the selective pressure is sufficient. If thousands of years of diving can produce detectable genetic change in spleen size, then millions of years in a semi-aquatic environment could plausibly have produced far more extensive anatomical changes in an ancestral hominid population. Where does the aquatic ape hypothesis stand today? Assessed honestly, and without the institutional defensiveness that has sometimes coloured the debate on both sides. It stands, as Elaine Morgan always said it stood, as a hypothesis rather than a theory, a proposed explanation that is consistent with much of the available evidence, but that lacks the direct fossil confirmation that would make it compelling to most specialists. The strong form of the hypothesis, that a specific semi-aquatic phase in the Miocene was the primary driver of the anatomical features that distinguish humans from other primates, remains unproven and probably unprovable with the fossil evidence currently available. The coastal and aquatic environments in which such a phase would have occurred are poorly represented in the African Miocene fossil record, and the absence of evidence, while not evidence of absence, creates an evidentiary gap that critics are entitled to find significant. But the weak form of the hypothesis, the claim that the human lineage has had a longer, deeper and more evolutionarily consequential relationship with water and marine food resources than the savanna theory acknowledged, has become increasingly difficult to dismiss. The nutritional evidence for DHA as a prerequisite for brain expansion, the archaeological evidence for early and extensive marine resource exploitation, the physiological evidence of the diving reflex and voluntary breathing control, the anatomical distinctiveness of human subcutaneous fat distribution, the rapid aquatic adaptation demonstrated in the Bajau, all of these converge on the same basic conclusion. Water mattered in human evolution. Not as a peripheral backdrop, not as an occasional resource, but as a defining ecological context that left permanent marks on the human body and brain. The debate about the aquatic ape hypothesis is, at its core, a debate about how science handles unconventional ideas proposed by people outside the establishment. Hardy's hypothesis was treated with contempt, partly because of its content, and partly because of who was defending it most loudly. A marine biologist and a television writer, neither of whom was a card-carrying member of the paleoanthropological establishment. The history of science is full of cases where correct or partially correct ideas were rejected for the wrong reasons, where institutional defensiveness and credentialism delayed the acceptance of valid insights by decades. The aquatic ape hypothesis may be one of those cases. It may not be. The honest answer is that we do not yet know, and that the question deserves to be held open rather than closed. The genomic revolution of the past 2 decades has given researchers tools that Hardy and Morgan could never have imagined, and the results have been, for the aquatic ape debate, genuinely illuminating in ways that neither side anticipated. If a semi-aquatic or consistently waterside phase shaped human evolution to the degree that the hypothesis proposes, we would expect to find in the human genome signatures of selection on genes related to aquatic function that are absent or less pronounced in the genomes of other great apes. And when researchers began looking for exactly those signatures, what they found was not a clean confirmation of the aquatic ape hypothesis, but something more interesting. A pattern of genomic differences between humans and other primates that is consistent with, though not exclusively explained by, a long history of water-associated selection. Human beings have significantly expanded gene families related to the metabolism of iodine compared to chimpanzees. Iodine is found in high concentrations in marine foods, particularly in seaweed, shellfish, and oily fish, and is essentially absent from the terrestrial plant foods that constitute the primary diet of forest-dwelling primates. The expanded iodine metabolism capacity of humans is a physiological adaptation to a diet rich in marine iodine sources, and it is not shared by our closest relatives. Iodine is also critical for thyroid function, and thyroid hormones play a central role in brain development and in the regulation of body fat distribution, which connects the iodine metabolism story directly to 2 of the most distinctive features of human biology: our large brains and our unusual fat distribution. The genomic evidence for enhanced iodine processing capacity in humans does not prove the aquatic ape hypothesis, but it is exactly the kind of genomic signature you would predict if our ancestors had spent a significant evolutionary period eating marine foods. And it is not the kind of signature you would predict in a purely terrestrial savanna lineage. The genetics of human skin and hair provide additional genomic evidence that is worth examining carefully. The molecular genetics of human hair loss has been studied in some detail, and the genetic changes responsible for reduced body hair in humans involve mutations in genes related to hair follicle development and the regulation of the hair growth cycle. What is interesting about these mutations, from the perspective of the aquatic ape debate, is that some of the same gene regions show signatures of positive selection in humans compared to chimpanzees, meaning that natural selection acted on these genes during human evolution to fix specific variants in the population. The presence of positive selection signatures on hair reduction genes is consistent with hairlessness being an adaptive trait rather than a neutral or deleterious one, which means something was selecting for reduced body hair in our ancestors. The savanna thermoregulation hypothesis provides one explanation for this selection. The aquatic streamlining hypothesis provides another. The genomic data cannot distinguish between them, but it confirms that reduced body hair was genuinely selected for rather than arising by genetic drift, which keeps both explanations in play. To understand what the aquatic ape hypothesis is really proposing and why it resonates with so many people who encounter it for the first time, it helps to look carefully at the mammals that actually did make the transition from land to water, and at what that transition left behind in their bodies. Because evolution does not invent things from nothing. It modifies what already exists, leaving traces of previous adaptations in the anatomy of organisms long after the selective pressures that produced those adaptations have changed. The bodies of semi-aquatic and aquatic mammals are, in this sense, geological records of their evolutionary histories. And when you read those records carefully, the patterns that emerge are instructive for thinking about the human case. Consider the hippopotamus, which is, in many ways, the most illuminating comparison for the aquatic ape hypothesis, because it represents a semi-aquatic lifestyle rather than a fully aquatic one, the same kind of lifestyle that the hypothesis proposes for ancestral hominids. Hippos spend the majority of their days in water, emerging at night to graze on land. Their bodies show a suite of adaptations to this semi-aquatic lifestyle that parallel, in striking ways, the features that the aquatic ape hypothesis attributes to water exposure in the human lineage. Hippos are hairless, retaining only sparse hair on the muzzle and tail. They have a thick layer of subcutaneous fat that provides both insulation in water and buoyancy. Their nostrils are positioned on top of their snout, allowing them to breathe while mostly submerged. Their young are born in water and can swim before they can walk comfortably on land. Hippos are the closest living relatives of whales, and their anatomy represents an intermediate point between a fully terrestrial mammal and a fully aquatic one. A snapshot of what a semi-aquatic transition looks like in anatomical terms. When aquatic ape proponents point to human subcutaneous fat and hairlessness as evidence of aquatic ancestry, they are pointing to features that are found in exactly this combination in the semi-aquatic hippopotamus, and arguing that the parallel is not coincidental. The fully aquatic mammals, the cetaceans and sirenians, show what happens when a lineage commits fully to the water over millions of years. Whales and dolphins retain vestigial hind limb bones buried deep in their bodies, visible only in dissection, remnants of the legs their ancestors walked on before the ocean claimed them permanently. Their front limbs have been modified into flippers, retaining internally the same bones as a human arm and hand, but enclosed in a streamlined flipper shape. Their nostrils have migrated to the top of their heads. Their body hair has been almost entirely lost, replaced by a thick blubber layer that is functionally identical to the human subcutaneous fat layer, but taken to a more extreme degree. The evolutionary process that produced a whale from a land mammal took approximately 15 million years, which is roughly the same timescale that separates modern humans from the common ancestor of humans and chimpanzees. The aquatic ape hypothesis does not claim that our ancestors went as far along the aquatic transition as whales did. It claims they went partway, far enough to acquire the fat and the hairlessness and the diving reflexes and the voluntary breathing control, but not far enough to lose their legs or fully commit to a water-breathing existence. The hippo, not the whale, is the anatomical model the hypothesis is built on. The return from water to land is, if anything, more interesting than the initial transition into the water, because it is the return that the aquatic ape hypothesis requires and that critics have found most difficult to accept. Can a lineage that has adapted to semi-aquatic life readapt to fully terrestrial life while retaining the aquatic modifications? The fossil record says yes, and says it repeatedly. The ancestors of modern crocodilians were fully terrestrial in the Triassic. The ancestors of modern penguins were flying birds in the Eocene. The ancestors of modern sea turtles walked on land. In every case,
The lineage moved into water, acquired aquatic adaptations, and then either stayed in water or returned to land with a modified body plan that retained evidence of the aquatic phase. The vestigial hind limbs of whales are the most dramatic example of retained aquatic evidence in a lineage that stayed in the water. What would the equivalent retained evidence look like in a lineage that went into water and then came back out? It would look like subcutaneous fat that no longer serves a thermoregulatory function on the warm savanna. It would look like a hairless body in an environment where hair would be beneficial. It would look like diving reflexes that are no longer regularly triggered. It would look, in other words, like a human being. The specific ecological scenario that the modern version of the aquatic ape hypothesis proposes is worth describing in concrete terms because it is more specific and more geologically grounded than Hardy's original vague proposal of coastal living. The scenario centers on the Afar region of Ethiopia and Eritrea during the late Miocene and early Pliocene, roughly 5 to 7 million years ago.
During this period, the Red Sea was in the process of forming as the African and Arabian tectonic plates separated, and the geological evidence indicates that the Afar lowlands were periodically inundated by marine transgressions, incursions of the sea into low-lying continental areas that would have created a complex landscape of islands, coastal lagoons, tidal flats, and shallow marine inlets. The Danakil Highlands, a raised plateau in what is now Eritrea and northern Ethiopia, would have been periodically isolated from the African mainland by this flooding, creating an island environment surrounded by shallow, warm water. A population of ancestral hominids trapped on the Danakil Highlands by the rising sea would have faced a specific and powerful set of selective pressures. The forest resources they had depended on would have been limited by the size of the island. The surrounding shallow waters would have offered an abundant and accessible alternative food source in the form of shellfish, fish, crabs, and other intertidal and shallow water organisms. An ancestral hominid that could wade into waist-deep water to collect shellfish from the substrate would have had immediate access to a food source unavailable to competitors that were unwilling or unable to enter the water. An ancestral hominid that could dive to retrieve shellfish from slightly deeper water would have had access to even richer resources. Over thousands of generations on a resource-limited island surrounded by shallow, productive water, natural selection would have acted on any trait that made individuals more comfortable in water, better able to exploit aquatic food sources, and more efficient at wading and diving. The subcutaneous fat that insulates against cold water, the hairlessness that reduces drag and makes swimming more efficient, the voluntary breathing control that extends dive duration, the diving reflex that protects the lungs during submersion, the upright posture that keeps the head above water while wading in deeper water. All of these traits would have been directly selected for in this environment, in this scenario, over this timescale. When the marine transgression eventually retreated, as geological evidence suggests it periodically did in the Afar region, the population that had spent thousands of generations adapting to the island waterside environment would have re-emerged onto the African continent carrying the suite of aquatic adaptations they had acquired. They would have been better bipeds than their mainland relatives because wading had selected for an upright posture. They would have had the voluntary breathing control that would eventually enable complex speech. They would have had a nutritional heritage of marine DHA consumption that had supported brain expansion during the island period. And they would have carried, in their bodies, the full set of anatomical peculiarities that distinguish modern humans from every other living primate. They would have been, in the most literal sense, the product of an island aquatic experiment conducted by natural selection in the shallow, warm waters of the Proto-Red Sea. And whether or not this specific scenario is precisely correct in its details, it demonstrates that the aquatic ape hypothesis is not merely a poetic metaphor, but a geologically specific and mechanistically coherent proposal that can be evaluated against the evidence as it accumulates. The evidence continues to accumulate. New fossil sites in the Afar region are excavated every decade, and the picture they are building of early hominid ecology is consistently one of waterside and mosaic environments rather than open savanna. New genomic studies continue to identify human-specific variants in genes related to aquatic function, iodine metabolism, fat distribution, and respiratory control. New physiological studies of diving populations continue to document the rapidity with which human bodies adapt to aquatic selective pressures. None of this evidence individually confirms the aquatic ape hypothesis, but collectively, it forms a pattern that is increasingly difficult to explain without reference to a deep and evolutionarily significant relationship between the human lineage and water. The question Hardy asked in 1960, sitting at his desk at Oxford with a list of anatomical curiosities that the savanna theory could not adequately explain, has not been answered. But it has become, in the 60 years since he finally found the courage to ask it publicly, a considerably better question. Perhaps the most provocative genomic finding relevant to the aquatic ape debate concerns the gene HACNS1, a human accelerated conserved non-coding sequence that shows the fastest evolutionary change in the human lineage of any regulatory element in the genome compared to the equivalent regions in chimpanzees and other mammals. HACNS1 acts as an enhancer, a regulatory element that controls the expression of nearby genes, and it influences the development of the limbs and, specifically, the feet and hands. The changes in HACNS1 in the human lineage have been interpreted primarily in the context of bipedalism and manual dexterity, the features most obviously related to terrestrial tool use. But the hands and feet are also the primary contact surfaces in swimming and in wading, and the anatomical changes in human hands and feet compared to chimpanzees, including the loss of the opposable toe and the development of the arch of the foot and the distinctive proportions of the human hand, are all changes that have been argued by aquatic ape proponents to reflect adaptation to wading and swimming as much as to terrestrial walking and tool use. The genomic evidence does not resolve this debate, but it confirms that the hands and feet were among the most rapidly evolving parts of human anatomy during the period when our lineage diverged from other primates, which is consistent with strong selective pressure from a new locomotor environment, whether that environment was a savanna or a shoreline. Beyond genomics, the cultural and behavioral evidence for a deep human relationship with water adds another dimension to the debate that is rarely discussed in scientific accounts. but that is anthropologically significant. Every human culture on Earth, without exception, has a profound relationship with water that goes beyond simple hydration. Water features centrally in the religious and mythological systems of virtually every human society, from the baptismal waters of Christianity to the sacred rivers of Hinduism, to the creation myths of Pacific Island cultures, to the flood narratives found in more than 200 independent cultural traditions worldwide. The universality of water symbolism in human culture is not by itself evidence for an aquatic evolutionary past, because water is obviously essential for life, and its symbolic importance could reflect its practical necessity rather than any evolutionary memory. But the specific quality of human attraction to water, the aesthetic pleasure that virtually all humans take in being near the sea or a river or a lake, the instinctive calm that most people experience when looking at moving water, the global phenomenon of coastal settlement that has characterized human civilization from its earliest expressions. These suggest a relationship with water that runs deeper than mere practical necessity. The therapeutic value of water immersion for human beings has been recognized across cultures and across history, and modern research has begun to quantify what traditional practices have long assumed.
Exposure to blue spaces, the term researchers now use for water-adjacent environments, including coasts, rivers, and lakes, has been shown in multiple studies to reduce cortisol levels, lower blood pressure, improve mood, and reduce symptoms of anxiety and depression in ways that are not fully explained by the general benefits of being outdoors or in natural environments. The specific quality of water, the sight, sound, and smell of it, has measurable physiological effects on the human nervous system that are distinct from the effects of other natural environments. Whether this reflects an evolutionarily ancient association between water environments and safety, food availability, and physiological comfort, or whether it reflects something more specifically aquatic in our evolutionary history, is an open question. But the consistency and universality of the human response to water across cultures, geographies, and individual histories is itself a datum that demands explanation. The peoples of the world who have historically lived most intimately with the sea offer a natural experiment in human aquatic adaptation that goes beyond the Bajau findings already discussed. The Moken people of the Andaman Sea, sometimes called sea nomads, have lived on and in the sea for generations, and they show physiological and perceptual adaptations to their marine environment that have been documented by scientific researchers. A study published in 2003 showed that Moken children have underwater visual acuity approximately twice as good as European children tested in the same conditions. Moken children who spent their childhood diving in the sea could see underwater with a clarity that European children could not match. When European children were trained to dive in the same way, their underwater visual acuity improved significantly, showing that the adaptation is partly learned.
But the baseline difference between untrained Moken children and untrained European children was larger than the training effect, suggesting a genuine perceptual difference rooted in developmental exposure rather than purely in practice. The Moken children's pupils constricted more in response to underwater light than European children's pupils did, a physiological rather than purely perceptual response. And this difference may reflect developmental adaptation during the critical period when visual systems are most plastic. The implication of the Moken visual acuity finding, taken together with the Bajau spleen enlargement finding,
is that human populations are capable of meaningful physiological adaptation to aquatic environments on timescales that are short by evolutionary standards. Both the Moken and the Bajau have lived their maritime lifestyles for centuries at most, not millions of years, and both show measurable physiological differences from neighbouring non-maritime populations. This rapid adaptive capacity does not prove the aquatic ape hypothesis, but it demonstrates with contemporary living populations that the direction of human adaptation the hypothesis proposes is genuinely possible and genuinely occurs. If a few centuries of maritime life can produce detectable changes in spleen size and underwater visual acuity, then the millions of years of semi-aquatic life that the aquatic ape hypothesis proposes would have been more than sufficient to produce the full suite of anatomical changes that distinguish humans from other primates. The question of diet and brain evolution deserves extended treatment because it has moved from a fringe argument associated with the aquatic ape hypothesis to a mainstream concern in nutritional neuroscience. The human brain is the most metabolically expensive organ in the body, consuming approximately 20% of total energy at rest, despite representing only about 2% of body mass.
Building and maintaining a brain of this size requires not just energy, but specific structural nutrients, particularly the long-chain polyunsaturated fatty acids that constitute the primary structural lipids of neural tissue. The most critical of these is DHA, docosahexaenoic acid, which makes up approximately 40% of the fatty acids in the brain's grey matter and which is essential for the development and maintenance of neural membrane fluidity and synaptic function. The human body can synthesize DHA from shorter-chain omega-3 fatty acids found in plant foods, but this synthesis pathway is inefficient, and the amounts of DHA produced from plant-based precursors are insufficient to support the rapid brain growth that characterized the evolution of the genus Homo. The preformed DHA that is directly available in fish, shellfish and other marine foods, by contrast, can be used immediately for neural tissue construction without the conversion bottleneck. The implication of this nutritional argument is straightforward and has been stated plainly by Crawford, Cunnane and their colleagues. The explosive brain growth of the human lineage during the Pleistocene was not nutritionally possible without consistent access to marine or freshwater DHA sources. A hominid population living exclusively on savanna foods, including terrestrial game, tubers, seeds and fruits, would not have had access to the preformed DHA in quantities sufficient to support the brain expansion that produced Homo sapiens. The consistent co-occurrence of early human fossil sites with lakeside, riverine and coastal environments is not a coincidence of preservation, but a reflection of a genuine nutritional dependency. Our ancestors lived near water because they needed to eat the things that live in water, and the brains that made us human were built, at least in part, from marine fat. This nutritional argument does not require a fully aquatic or semi-aquatic lifestyle, in the sense that the original aquatic ape hypothesis proposed. It requires only that our ancestors had consistent access to aquatic food sources over the extended period during which brain expansion was occurring. Lakeside and riverine environments would have provided sufficient aquatic food resources without requiring swimming or diving, and many researchers who reject the aquatic ape hypothesis in its stronger form accept the nutritional argument for waterside foraging as a significant factor in human cognitive evolution. The waterside hypothesis is, in this sense, a minimum version of the aquatic ape insight
that has achieved substantially more mainstream acceptance than its more dramatic predecessor, precisely because it makes a more modest and more directly evidenced claim. There is an image that Elaine Morgan returned to repeatedly in her writing, an image that she found more persuasive than any anatomical argument. It is the image of a human infant placed in water. Not a trained infant, not an infant that has been taught to swim, just a very young human baby, a few months old, placed in warm water with its face submerged. And what happens next? The heart slows. The tiny glottis closes. The small arms and legs begin to move in a coordinated paddling rhythm. The infant does not panic. It does not struggle. It performs without instruction, without practice, without any learned response, a behavior that is not present in any other infant primate. A behavior that is present in the infants of diving mammals. A behavior that looks, in the context of everything else we know about human anatomy and physiology, like a memory. Not a cognitive memory, not something stored in a mind, but a biological memory, a pattern written into the human body by an experience so deep in our past that it preceded language and tool use and fire and everything else we call human. A pattern that says, in the body's own language, we have been here before. We know this. The water is not strange to us. We remember it. Whether that memory is what Alistair Hardy thought it was, whether it dates to a specific semi-aquatic phase in the Miocene forests of the Afar, or whether it is something older and more diffuse, a general primate relationship with freshwater and coastal environments that shaped us less dramatically but no less permanently, is a question that the fossil record has not yet answered. But the memory itself, written in the reflexes of every human infant who has ever been placed in water and performed, without being taught, the motions of swimming, that is real. It is there. It is you. And the fact that no one has yet produced a completely satisfying explanation for why it is there is, in itself, the best argument for keeping the question open.